Welcome to Assay Genie. In this video, we will deep dive into our membrane antibody arrays. We will explore how these powerful tools can revolutionize biomarker discovery, drug response monitoring, and disease research. Biomarkers provide crucial information about molecular pathways in diseases. They help identify the most suitable drugs, determine the best sample type, and gain insight into drug mechanisms of action. With the right biomarkers, drug discovery and treatment optimization become much more effective. Biomarker discovery plays a vital role in precision medicine. The ability to measure biomarkers across multiple sample types, blood, urine, saliva, and tissue extracts opens new avenues for personalized healthcare. By identifying biomarkers linked to specific disease pathways, researchers can design targeted treatments that improve patient outcomes. Despite the potential of biomarkers, researchers face significant challenges. Time-consuming and costly validation processes slow down drug development. Limited high-throughput analysis options restrict the ability to analyze multiple biomarkers at once. Sample volume constraints and inconsistent data reproducibility impact research reliability. But what's our solution? Multiplex antibody membrane arrays. These allow for the detection of 7 to 274 proteins per sample, analyzed using simple yet advanced technology. This innovation streamlines biomarker discovery and validation. By incorporating high-throughput multiplexing, these arrays enable researchers to capture a more comprehensive picture of disease-related protein expression. With validated antibodies used throughout the discovery workflow, the process follows as a structured approach. Discovery, identify potential biomarkers with Assay Genie's membrane antibody arrays. Verification, validate biomarker presence in disease samples. Selection, choose relevant biomarkers for further research. And qualification, ensure biomarkers meet criteria for diagnostic or therapeutic use. This workflow ensures accuracy and reproducibility in biomarker analysis, reducing false positives and increasing confidence in research outcomes. What's included in the kit? Antibody arrays, booking buffers, biotinylated antibody cocktail, and more. Additional items you will need: pipetors, pipette tips, an orbital shaker, blotting paper, distilled water, and a chemiluminescent detection system. Having the right laboratory setups ensures optimal performance of your antibody arrays. Proper handling and precise pipetting techniques further improve data accuracy and reliability. Here's a step-by-step overview of how membrane antibody arrays work. 1. Prepare your sample. 2. Apply the sample to the membrane. 3. Incubate with detection reagents. 4. Wash away unbound material. 5. Detect proteins via chemiluminescence. Our arrays provide high-quality, reproducible data using chemiluminescent imaging. Positive control spots ensure consistency, negative control spots measure background noise, and blank spots confirm accuracy. Data can be analyzed using widely available software, making it accessible for researchers worldwide. The ability to perform comparative densitometry allows scientists to quantify protein expression differences between samples, providing valuable insights into disease progression and treatment efficacy. We ensure all array targets are fully validated with Genie ELISA and Western blot antibodies. This guarantees the accuracy and reliability of the data collected using our antibody arrays. Let's look at a real-world example. Researchers needed a technology to screen multiple targets simultaneously for obesity-related studies. Our membrane arrays allow for the detection of 62 adipokines in treated versus untreated cells. Researchers could analyze multiple biomarkers in one experiment, saving time, reducing costs, and increasing accuracy. This case study highlights how multiplex arrays can accelerate discoveries in metabolic and obesity research. By using our arrays, researchers could efficiently detect relative differences in 62 adipokines using simple instrumentation. Short protocol, low cost, high reliability, a game changer for obesity research. To wrap up things, here's why our membrane antibody array stands out. Dynamic range, detects protein levels over a wide range, exceeds needs, over 100 antibody arrays available, high sensitivity, capable of detecting as low as 1 picogram per milliliter, compatible, works with any chemiluminescence imaging system, and peace of mind validation. Arrays are validated with Genie ELISA. Thank you for watching. To learn more, visit our website or send us an email at info@assaygenie.com.